Okay, let's get started. Thanks everyone for joining on time to today's Open Box Science, Genomics, and AI webinar. And today we're very excited to have Ben Oromega, and he's a biologist graduated from Uruguay's Public University and trained in AI for complex disease prediction and genetics at Mount Sinai. He's currently a PhD student and Clauderian Scholar at the University of Oxford's Deep Medicine program, developing AI tools to enable remote cardiovascular cares in rural regions of low-income countries. And a fun fact about Ben is that he studied biology thanks to a guy he met in a hostel in New Zealand and don't even remember his name. So wow, what a journey you have been on. So Ben, take it away, please.
Okay, thank you for the introduction and thank you for um, giving me the space to present our latest work with Rondeau and the lab at Mount Sinai. Here we did an exome analysis on coronary artery disease using a machine learning-based phenotype. And so a bit of background. As some of you may know, coronary artery disease or CAD is a very well studied disease genetically. There have been very large scale genome-wide association studies now of over 1 million participants looking at the genetic architecture of the disease. But the issue with genome-wide association studies is that they mostly identify common loci, which don't directly inform the role of a gene in disease biology. And for that, we are more interested in rare coding variant associations. Rare coding variants directly inform causality of a gene in disease biology because we assume that the rare coding variant disrupts gene function. So the association can be seen as a proxy to the role of the gene in disease. But for coronary artery disease, opposed to genome-wide association analysis, exome-wide association analysis haven't been so successful in the last decade. These are the three papers that we could identify in this last decade, having found novel genes uh, linked to CAD with rare coding variants. And compared to the 321 genes that we saw earlier from that review, we only could find 4 genes linked to CAD in the last decade with rare coding variants. And so why is it so challenging to identify novel rare coding variants for CAD using the traditional phenotypes of CAD cases and controls defined based on diagnostic codes? So we can think of 4 main challenges and reasons. So the first one being intrinsic to studying rare coding variants. These are defined as having a minor allele frequency lower than 1%. And so the low number of carriers in the analysis um, lowers statistical significance of the association test. It makes it harder to identify significance in the signal. But then also the use of diagnostic codes has known biases and can result in misclassification, which at the end introduces noise into the test statistic and also reduces power to identify significance. An issue intrinsic of studying CAD, it has low prevalence, around 6%, I think. And so again, the number of carriers with the disease is even lower, and so it's harder to identify significant genes. And then the use of cases and controls. It's problematic for CAD in particular because cases and controls represent disease onset, so the diagnosis of the disease, but are not able to capture other axes of disease pathophysiology involving things like progression to disease or severity of disease, which is very important in CAD. You have individuals that are more severely affected by atherosclerosis. But then also things like underdiagnosis. So in CED, it's not uncommon to have individuals that present some kind of atherosclerotic burden in their arteries, but haven't been yet diagnosed formally with the disease. And so you again have noise in the association statistic when you consider all these individuals as controls. Um, so 3 of the 4 challenges involving rare variant discovery in CED refer to issues with phenotype and suboptimal phenotyping of the disease. So we thought of asking the question, how can we improve phenotyping of CED to potentially reveal novel rare coding variants for CED? And so in this line of thought, our lab had already published a paper in 2022 showing that machine learning models can learn from structured electronic health records to accurately predict coronary artery disease. And so in essence, what we did was train simple tree-based models to learn from widely available structured EHR, and these involve vital sign measurements, laboratory test results, diagnostic codes, and medications. And the model showed very good prediction of coronary artery disease in terms of discrimination or AUROC. We found in an external testing dataset within Biomi, which was the training cohort, 0.94 AURC and precision of 88%, but then in a completely external cohort, population-based, mostly healthy individuals, so very different to Biomi, we also found good discrimination at 0.88 and good precision at 92%. We compared the performance of our machine learning tool to widely used risk prediction methods such as the pooled cohort equation and also under investigation polygenic risk scores. And we demonstrated that the machine learning based tool could improve prediction by 12% in terms of AUC and 14% in terms of precision. So very good performance, and amongst all, I think it's a reason to believe that machine learning can find signs of disease biology in the EHR of our cohorts, and so a way to capture disease biology for CED in particular. And that's what brought us to think that this same methodology and approach could be used to characterize CED. There have been studies showing that CED, as opposed to other diseases, exists on a spectrum, so there are different gradients of atherosclerotic burden that lead to different outcomes in a scale. And so we thought of this is work from Ian Forrest, an MD PhD student at Ron's lab. And the goal here was to try to develop a non-invasive in silico tool to measure this spectrum. And the analysis showed that the machine learning based tool does a very good job at tracking different signs of disease biology and can quantify atherosclerosis on a spectrum as well as risk of mortality from CED. So I'm giving here a QR code if you want to see the analysis in depth, because there's a whole lot of interesting analysis that I'm not going to go into detail here, but are important for the next paper that we're going to discuss. So I encourage you to read the QR code and see the analysis. But in essence, What the paper shows is that this ML tool can track progression, severity, and underdiagnosis of CAD. So when we compare traditional phenotypes that we use for genetic associations of cases of control that we are going to refer as CAD status with this novel in silico-based tool, ISCAD, we can say that CAD status only captures disease onset, while ISCAD captures disease onset, but then also progression, underdiagnosis, and severity. So potentially a more holistic representation of CED that is closer to the actual spectrum of CED that we know exists. And so we thought whether this more holistic phenotype is a way to improve phenotyping and then therefore genetic discovery in CED and tackle this big issue of identifying rare coding variants for the disease. So onto the methods of the paper, how we came on to test this. We took EHR and genetic data, both non-imputed genotype data and whole exome data from 3 independent cohorts. There is the UK Biobank, our largest cohort, but then also the All of Us research program in the US and Mount Sinai's Biobank. In total, we computed ISCAD and tested genetic associations in over 600,000 individuals. I'm going to go through the workflow with these little icons. So we identify as traditionally CED cases and controls, and we use those to train the machine learning models. The machine learning models learn from, as I previously mentioned, vitals, labs, diagnostic codes, and medications. And these are then used to compute ISCAD on a spectrum, goes from 0 to 1, 0 being less atherosclerotic burden, 1 being more atherosclerosis in individual, in each independent cohort. So we trained 3 different models, 1 in each cohort, and computed the score in each cohort, and then also run genetic associations in each cohort to then perform a meta-analysis of the association statistics. So we tested rare coding variants in 3 different ways. First, single variants. We tested rare coding variants, as I mentioned before. These are variants with less than 1% allele frequency, but then also we tested ultra rare coding variants, which have less than 0.01% frequency in each cohort. As coding, we refer to variants that are missense or protein truncating variants. Then we also tested gene level aggregates of deleterious ultra-rare coding variants. And for this, we took ultra-rare coding variants that have either less than 5 counts in the minor allele within each cohort or less than 0.001 allele frequency in each cohort. And these have to be protein truncating variants or missense variants with 5 out of 5 pathogenicity predictions in 5 independent computational tools. So we aggregated these highly deleterious variants per gene, and we tested almost 18,000 genes separately. And then additionally, we also tested biological processes. For this, we took 50 hallmark processes from the MSeq database, and these basically aggregate genes in each process. So we took the same ultra-rare deleterious variants, gene level ultra-rare deleterious variants aggregates to build these 50 gene sets of biological processes and tested those for genetic association with ISCAD. We used Regini for all of our association tests and also performed associations with cases and controls, so the traditional phenotype for CD in all tests. for comparison. And then finally, this bit here was a very important part of the work because our results are associations with ISCAD and not with traditional CAD diagnosed individuals. And so it was very important to characterize and understand the role of these genes in disease biology to ensure that our associations are not artifacts of the score and possibly non-specific to CAD as the score is training all of the EHR, but rather genes that are specific to CAD biology. And so we assessed the role of these genes in CAD from different angles. We tested them. I'm referring to only the significant ones, right? So the genes that were significant in association with ISCAD, we tested them for association with clinical manifestations of CAD, such as arterial stiffness index, ejection fraction, and comorbidities, including arrhythmia and stroke, among others. But then also we evaluated their association in previous GWASs of common variants, but only looking at coding variants and EQTL loci. We assessed their involvement in CED using a review that came out in 2019. And then also we looked at where any of these genes have been previously tested as a drug for CED in clinical trials or have rare coding variants associated with it. So we assessed the role of these genes from many angles. And so into the performance of the predictive model. We found very good discrimination for the three models in 3 biobanks, 81 in the UK Biobank, 86 in RFS, and 89 in Mount Sinai Biobank. Then I think it's really interesting to see how the distributions of the different scores differ between cohorts. We see bimodal distribution for example, the all-of-us model in the controls, and then also the BioMe model tends to overestimate risk in controls. So very different distributions between the three models, which I think is important because we don't really understand and we don't really know what features of the ML model are driving the genetic associations. One would expect that a model that performs better in terms of prediction would give better genetic associations or closer to reality genetic associations, but we have some secondary analysis in the paper showing that this is not the case. If you think about it, a model that has perfect predictive performance would be equal to testing for association with cases and controls. And so we are actually trying to complement cases and controls. So we are not looking for a model that has the best possible performance. So how does distribution impact the genetic associations? Is it important to determine which genes are going to be associated with the score? Or could it be the features that drive the prediction? So in that sense, we also assessed the feature contribution for each of the models. And we have some secondary analysis in the paper showing that causal risk factors may play an important role in identifying disease mechanisms that are involving traditional risk factors for CAD. So amongst the features that drive at least 50% of the prediction in each cohort, we see using the asterisks many causal risk factors involved in the prediction in all of the cohorts. So again, this is an ongoing line of research, understanding what features of the model drive genetic association, which I think is very interesting. So onto the...
so sorry to interrupt.
Yes. No worries.
Sorry. Sishank. Hi. Could you
Yes.
describe the training process for those models on the previous slide, please?
Yes. Okay. So the models use structured EHR, and that includes vital measurements, laboratory test results, diagnostic codes, and medications. You can divide those into continuous data and categorical data. Labs and vitals would be continuous. So for those, we scale the data. For categorical data, we perform feature selection within each of the two classes, and then we throw that into a tree-based model, a random forest model, and let the
What was
model.
the class break? Was it like a pretty even class breakdown between the control and CAD cases across the three data sets?
Right. We train the models using a balanced set. So for the CAD cases, we take all EHR up until their diagnosis. For controls, we take all of their EHR with no masking date. And we take 90% of all of the available cases, CAD cases, and an equal number of controls. So if 90%, it's 9,000 CAD cases, we also take 9,000 controls and train a model with that. And we do that 100 times
Got it.
to randomly sample different individuals for the training and then leave that 10% out for testing. And with those 100 models, we predict 100 times in all individuals and average the predictions to get the final like ISCAT score in all
Okay.
individuals.
Thank you.
Yeah. Okay, so with those models, we go into the association. We tested record invariance for association, and we found in single variant analysis 12 significant hits. Some of these might pop up as well-known CAD genes, which are important for positive controls, such as ApoB, ApoC3, more controversial genes such as LibG. But I think it's really important and interesting how a third of the hits, so four variants, replicate between the different cohorts, so between the three association tests that we perform. And then when comparing the associations with CAD status, we see a six-fold increase in significance. This is only in genes or variants that were significant for ISCAD. So there's still the question remains as to whether this increase in significance can be applied to or transferred to any CAD-linked gene or just to genes that are significant for ISCAD. But still, interesting preliminary results to see such an increase in significance. And then also six of the variants, so half of the variants are protective for ISCAD, which could inform therapeutic targets or disease mechanisms that can reduce the risk of CAD. And then onto the gene level associations, we found 7 significant genes. 4 of them, so more than half of them, replicate between cohorts. That's very important. And as well, we see well-known CAD genes such as LDLR. This was, as we discussed earlier, discovered in 2014. It's a very well-known gene for CAD, ABCA1, ApoB again, and then also other genes that involve traditional risk factors such as diabetes, GCK, and PPARG. So interesting results. And onto the biological processes, out of the 50 processes that we tested, 7 came up as significant. Some of them are well known for CAD, such as pancreatic beta cell function and transforming growth factor beta signaling. But also there are other ones that haven't been described for the role in CAD before and are pretty interesting, such as protein secretion pathways and the progression throughout the cell division cycle. as I mentioned, an important part of the paper was trying to understand whether ISCAR associations are specific to CAD. And for that, we tested ISCAR and CAD status for association with very well-known genes for CAD. These are 7 independent genes, and we show that in variant level analysis, 6 of the 7 variants show stronger or similar association with ISCAR compared to CAD status. And in gene level analysis, that's 7 out of 7. So all of the genes that we tested show similar or stronger association with ISCAR, suggesting that ISCAR is capturing CAD specific associations. The one variant that we didn't find association with ISCAR and is very well known for its role in CAD is PCSK9. And for this, we have a small discussion in the paper. So in total, we found 17 genes associated with CAD, recording variants in 17 genes, because our associations are with ISCAR and not with traditional diagnosed CAD cases. Rather than trying to label which ones are novel and which ones are known, we built this tier system where we classify them based on the support for the role in CAD. And so for that, we investigated many lines of evidence from stronger support to less strong or weaker support. The genes that have been tested for drugs in clinical trials or have rare coding variants are very well known genes for CAD. Then genes that have some kind of genetic support, mostly from common variants in GWAS, or have been highlighted in this recent review that I mentioned earlier. These are tier 2 genes. 8 of our 17 genes are tier 2 genes. So you could say have genetic support for the role in CAD. Tier 3 genes don't have genetic support, but have clinical support. We tested this for association with clinical manifestations of CAD, as I mentioned, arterial stiffness, index ejection fractions, among other, and their association with causal risk factors. All of these 3 genes showed a role or at least an association with these outcomes. And then tier 4 genes have none of the above, but have common variant association with causal risk factors. And then also literature review of all of these genes that we show in the supplements show some lines of support when you look at the biological mechanisms of these genes. So OSBPL3, for instance, it's involved in oxysterol transportation, which oxysterols play a role in the oxidation of LDL during plaque formation, so could be involved in disease mechanism. And then EML3 plays an important role in microtubule assembly, I think. And that's a really novel line of studies looking at microtubules for therapeutic targets of CAD. So again, some signs of role for these genes in CAD. And if we go into detail with some of these others, I think it's interesting to see in tier 3 genes, PLCB3, very well known for its role in hypertension, PPARG, as I mentioned, in diabetes, and SOS2, this came up as protective for CAD and has been, or is actually very well associated with Noonan syndrome 2, and its protection against Noonan syndrome 2, a monogenic disease that causes malformations of the heart. Then tier 2 genes involve anything from genes that have a role in diabetes to genes that have a role in apoptosis, such as MCL1, and this gene is being tested for clinical trials as a target for cancer. And LIP G, this is a very controversial gene in CAD. There are papers showing that it does increase HDL cholesterol, but has no effect on CAD onset itself. So a lot of things to unpack here. And then ApoB, ApoC3, and LDL are just very well-known gold standard genes for CAD. In total, 14 of the 17 genes, 82% have moderate, at least moderate support for the role in CAD, which is important to be sure that the associations with ISCAD are specific to CAD and represent CAD genetic architecture. And then also, I think very, very interestingly, from tier 3 and tier 4 genes, so genes without genetic support, none of them regulate lipid pathways. So a lot of the genes that have been identified using GWAS rely on cases and controls, which came up to discover a lot of genes that regulate the metabolism of lipids, but nowadays we know that CAD is not only restricted to the metabolism of lipids, so would look like these tier 3 and tier 4 genes could shed some light into the non-lipid pathways of CAD. So in conclusion, ISCAD was associated with rare coding variants in very well-known CAD genes, so definitely capturing CAD biology, but then also novel or not known genes for CAD, which represent horizontal biological process not only restricted to traditional risk factors and lipid pathways. So in this sense, you could say that genes associated with this can capture additional access of the CAD pathophysiology beyond CAD cases and controls, which you could think are skewed to the disease mechanisms that are used for disease diagnosis, which isn't the case with ISCAD. ISCAD considers all of the EHR. And open questions are whether ISCAD actually increases statistical power altogether, and that's why we identify rare coding variants, which were lacking in the last 10 years, or does it identify novel pathways that you cannot identify with CAD cases and controls and serves as a complementary tool. And then also understanding what features in the model drive genetic association. That's very important to understand what's the disease mechanism that you are capturing with the score. And so, as I mentioned, this study was published this year in Nature Genetics, and giving the QR to a PDF of the paper if you want to access and give it a read. And a big thank you to all of the team for making this possible. When I joined the lab, I learned a lot from these people. First, Professor Rondeau, my advisor, he came up with the idea of using ISCAD for genetic association and guided the research throughout the process. Ian Forrest, the medical student that tested ISCAD, many clinical outcomes, and all of the team in DOLAB, which supported the study, and then our collaborators, mostly clinical collaborators, such as Dr. Rosenson and Dr. Malik and Una Wardena, very important for their support. And thank you everyone for listening.
Awesome. Anyone feel free to raise your hand if you want to unmute and speak your question, or you can post on the chat. So I can start by a couple of questions. So I'm a little surprised by, okay, I'll ask Hari after. I'm a little surprised there's only 4 previously identified, because I imagine CAD being so common, the case control studies in the past has been quite big, and when they recruit case and control, they probably won't need as much of an SCAD approach because hopefully they identify really strong cases and controls. So how big was the previous biggest X and Y association study, and why do you think they only found four?
Um, I think the largest exome-wide association study to date is the one from 2014, which is actually work from Rondeau, PI. And I don't remember the numbers, but it's significantly less than what we tested, which is over 600,000 individuals. There have been only 4 novel genes identified through exome-wide association analysis, and that's because exome-wide analyses are agnostic and they are not targeted towards a particular processes. I think there are other papers that have identified rare coding variants for CAD, but in a more targeted approach. So not in exome-wide analysis altogether, but looking at a particular genotype, so a particular gene, testing that with a larger sample size, for instance. So I think in exome-wide analysis altogether, there haven't been so many successful studies because it's an agnostic test and you need a very large sample size to identify some significance.
I see. Okay. Harry, can you ask the next question? You can unmute.
Sure, yeah. I mean, thanks so much, Ben. That was such a great talk. So I actually have a couple of questions. One, when you conducted those burden tests in biological processes, did you basically, for example, say if there are 100 genes, they're part of a particular biological process, there's going to be some genes that activate the process and some that inactivate the process. Did you split them up separately and then conduct burden tests?
Splits what separately? Sorry, I missed that part.
So, I mean, if there are like 100 genes, for example, in a particular pathway, say half of them try to deactivate the pathway, right? And then the other half try to activate the pathway, right? Genes that inhibit or activate the pathway. When you conduct burden tests, we can't really put all those 100 together, right? We should just look at those, I mean, collapse all the genes that try to deactivate the pathway and then conduct a burden test there. And then those that activate the pathway, you can probably separately do a burden test in that gene set rather than like combining everything into one whole set. I was just curious if you consider that at all.
That's a very good point. We didn't test them separately. So the database that we took is the MSeq database, and it just gives you the genes that are involved in the pathway. It doesn't separate them between activating and deactivating. That might actually be introducing noise into the test and reducing significance. So that's a very good point. We just included all of the genes that we know have a role in the pathway.
Gotcha. Gotcha. So you didn't apply SCAT tests here, right? Probably SCAT tests would be a little bit more powerful, I guess, when you have genes going up and down in a given pathway.
Yes, we tested SCAT, ACAT, and burden.
Okay.
And we took the one that gave the stronger significance.
Okay.
For the direction of effect, we only considered the effect from the SCAT test. But yeah, again, if we combine both the ones that activate and deactivate, then I think the direction of effect is going to tend to no.
Got you. Cool. So another thing I was just curious, were you surprised to find only 17 genes with like 600K samples? You know, you ran both burden tests, CAT tests, and then you're using essentially like a continuous phenotype. Were you kind of surprised or just happy that you found 17?
No, I think it's a huge number. There have been there is one study, I think it's not published, so in a conference where they did a rare coding variant association with cases and controls in the UK Biobank. So something above 450,000 individuals, and didn't find any novel genes. They just found the very well-known genes. So having identified novel genes and known genes, I think it's very strong. We also tested different ISCAD scores and with different, it's on the paper, but different restrictions and filters in the training on the model. And this one, which doesn't have any kind of filtering, was the one that yielded more associations. So I think if you see as these rare coding variants, some of them having less than 10 carriers, the power to find significance in those variants less than 10 carriers is really low. So having identified 17, I think is pretty strong.
Gotcha. And then maybe one last question I want to ask. So if you can go back to the feature contribution slide. The second column that you have that says causal RF, and then you had asterisks. Are those like features that we know based off of domain knowledge that are on the causal path to
Yes.
CAT, or is that what's going on here?
We defined, I think it's 6 or 7 risk factors that, thanks to our clinical collaborators, they label as causal. So an increase or decrease in those features would mean a decrease in the risk of having the disease. So these are features that involve like lipid pathways, LDL, diabetes, hypertension, triglycerides, again, lipids, and BMI. I think that's still weight. BMI is still one. So yes, these are supposed, these are causal for the disease. We know that a change in these features would mean a change in the risk for the disease.
Gotcha. And then here you're basically showing all the features that you used as inputs across these 3 data sets, or were these sorted based on their contribution scores?
Yes, these are sorted based on the contribution. So aspirin, H, and H are the most heavily contributing features. I'm only showing up until the features that make up to 50% of the contribution in the model. I think the number of features is well above 100 in all of the models. And here we are only looking at a subset which heavily contributes to the disease, and within that subset, how many are causal? That's the question.
Gotcha. Thanks.
Or people have other questions. I follow with the technical question too. You require the variance to be 5 predicted for missense, I presume, by 5 different tools. Have you tried different? Is there a reason for using 5?
That's from Ron's Nature paper on where they discovered LDLR and ApoA5. They came up with this approach only for gene burden testing, right? In single variant, we don't restrict to variants that are deleterious in these 5 out of 5 scores. But because they were looking, they were trying to build gene sets that are highly deleterious, they came up with this approach of using 5 out of 5 pathogenic deletions in 5 very widely used scores. So we replicated that approach. Now, shortly after we did this analysis, there was a very strong, I can't remember the name, but a very strong tool that came up for missense variant prediction, pathogenicity in missense variants. I think it came up in Nature. So we are actually using pathogenicity prediction just in that one score now. But this comes from the paper that Ron published in Nature.
Got it.
I have a question about the AUROC image that you'd shown comparing different model training sets. That's the one. Yeah. So why do you think it is that when trained on just a polygenic risk score, the model performs worse, but with the EHR data and polygenic risk score, there's no improvement compared to just EHR data. Is there like feature or I guess data redundancy in what's captured there?
Yes, and mostly would think of that of the added value of the PRS already being captured by some of the clinical features. And I think most of it comes from covariates, age and sex, and some other that are also tested in the PRS score. So when we say PRS here, it's PRS plus age and sex. So yeah, it would be that the value of the PRS is already captured by clinical features in the EHR.
Gotcha. And do you know if that PRS contained any genes, I guess, that are associated with some of those causal risk factors that you mentioned later on?
The PRS thing is built on all of the associations, but they are common variant associations. So I think it's like a different realm of things. The betas that you use to compute the PRS are not really causal for the disease, whereas the rare coding variant, the betas from the rare coding variants are much more causal for the disease.
Thank you.
Anyone else have any last questions for Ben? Okay, if not, thanks everyone for listening, and importantly, thanks Ben for presenting his work. That was really, really helpful, and hope to see you next time. Bye.
Thank you for the opportunity to present. It was very nice.
Of course.